you for inviting me to reach in, and I'm going to give you guys a quick talk of what I can get released for a presentation for this meeting. I work at something called the National Biodefense Analysis and Countermeasure Center. It is a place where we look at forensic samples where people do very unintelligent things and mail things that they shouldn't mail, or make things that they should not make. So what I'm going to be talking about is imaging hazardous organisms, the procedures, protocols, and compliance for biological select agents and toxins. And as I go, I'm just going to pause and tell you things and just try to give you an inclination of what we actually do, and try to tell you what I do without telling you what I do, if that makes sense. Okay, so this is the building I work in. It's a national laboratory for the Department of Homeland Security. And we have biological safety level laboratories at biological safety level 2, 3, and 4. So you see all those guys in like those blue suits or white suits. Well, we do actually wear those, and we do process EM samples at BSL 2, 3, and 4. Okay. So if anyone has ever seen me give a talk, I always show this slide. Okay, this is my favorite slide. I took this slide in approximately 1996. This is using an antibody against a protein called supervillain that I cloned. And this is a single tissue culture cell here. And this is the nucleus of the cell, and this is the endoplasmic reticulum around the cell. And this protein, supervillain, is actually a membrane-bound protein, and it should be localized out here on the plasma membrane. And we made several different antibodies against this protein, but this one that gave beautiful endoplasmic reticulum staining here is actually simply artifact. So what as a microscopist, you have to have a good background in what you should see to understand what you're actually seeing. Okay. So what we're going to talk about today includes more things like this. Okay, bioforensic electron microscopy. We've looked at lots and lots of things like this. This is actually an anthrax spore. And this is a scanning electron microscopy image, and this is a transmission electron microscopy image, and then they're pseudo-colored in Photoshop. So if you don't like the colors, you can blame me, because I make lots and lots of Photoshop images of pretty microscopy images. So the image on the left, this is actually a single anthrax spore, and you ask yourself, what information can I get simply by looking at this spore? Information you can get: does it look like it's a pure culture? Does it look like it's mixed with something? And if it's mixed with something, can you key out what it's mixed with? This spore is simply sitting on a membrane filter with a 0.2 pore background. You can do elemental analysis if it's mixed with something to help figure out what it actually is mixed with. Does the spore look like it's been tampered with in a certain way to make it more nefarious? Okay, so we look at lots and lots of these kind of spores. We've looked at anthrax spores and all bacillus spores and most spore-forming organisms, to get a database of all organisms, so that when we get unknown samples, we can say these samples are consistent with the images that we have of these anthrax spores. And you're taking my word for it that this is an anthrax spore. Okay, so one thing that we did when we set up our facilities was we procured known traceable samples of all hazardous organisms, and we imaged them the exact same way. So that when I tell you this is actually a picture of an anthrax spore, I actually have a certificate of authenticity and proof that what I'm showing you is actually what we have. Okay, so for all hazardous organisms, we have all these images, and we have all these certificates, and we have proof that when we show you something, it actually is what it is. So what do we do? Okay, so we get evidence from numerous different clients. Okay, so if you think of those federal agencies that do any kind of police work, we get evidence from those. Typically, we get evidence that's biological in nature. So if someone thinks that there's a biological agent in it, we will get that evidence so that we can image it, and we can inactivate it, which we're going to talk about complete inactivation protocols in a few minutes. We have to inactivate it and prove it's inactive before we can actually image it to help say what it is. Okay, so there's evidence that goes through agent ID, but it goes to bacteriology, it goes to virology, or to toxinology, or if it's mixed, it goes to several different places. But down here, this is the EM lab. We do physical, chemical, and biological analyses to help figure out what might be there. And all this leads to what is called data for attribution investigations, which means it's data for getting search warrants and things like that so that you can arrest people and prosecute them. There's also classical forensic data that numerous clients will do to get fingerprints, trace fiber analysis, fire marks, and tool marks and firearms, and other kinds of traditional forensic data. So what do we look at? Types of samples that we get. We get purified bacterial cultures from the bacteriology department. We get purified viral cultures from the virology department. We get all kinds of toxins from the toxinology department. We get solid surfaces, such as this guy down here. This is a piece of a pipe, okay? This pipe down here, we actually did elemental analysis, and we determined that this is actually was supposed to be a specific kind of pipe. And with elemental analysis, we were able to figure out this was a counterfeit pipe. This type of material was a slag material that was never made into a pipe. So someone was using this slag material from the 1970s, making pipes with it and selling it when it should never have been used, okay? So we look at lots and lots of different kinds of things. We look at aqueous solutions. So if we get anything like an oil or a liquid or a water, we have to figure out ways to look at these samples. We get dry specimens like cakes and powders. And one of my technicians said I should get like a lyophilized cupcake pitcher and put here. So it's a very dry cake, okay, or powder. I thought it was funny, but no one else does, but that's okay. We also get dry specimens that are water soluble. And this is a problem for us, these water soluble specimens, because when you get an unknown sample, you have to know how to look at it. So if you were to get a water soluble sample and you put it in traditional aqueous fixative, it's going to dissolve. Hence, you've just lost your sample, which means that you've lost any evidence that you had. So we have to be able to look at types of evidence where we can look at it without anyone knowing that we actually looked at it. Okay, so we have certain samples that we look at that we can't have any markings or any implication that we've actually looked at those kinds of samples, because they have to go back sometimes to the actual clients. So if these are actually Bacillus anthracis bacteria that make the anthrax spores. I'll point out things as we go along. Okay, this was actually a piece of pipe. This is a cell pellet that we got one time. This is actually a salt solution we got one time. You can see where it crystallized and dried. And this was just a comparison so that I could show this what rice looks like compared to anthrax spores one time for a client. So inactivation and preservation. And we heard in the previous talk, we talked about lots and lots of preservation, okay? In my field of work, it's mostly about inactivation, because I'm working with agents that cause disease, extreme disease, okay? So common fixatives that we use paraformaldehyde, glutaraldehyde, mixtures of paraformaldehyde and glutaraldehyde, and osmium tetroxide. And the reason we use these is to cross-link the cellular structures and to inactivate the structures, because what we have to do is we have to be able to inactivate these things and get them out of a BSL 2, 3, or 4 laboratory for us to image. Unlike, say, a hospital or a university laboratory, we actually work with things that are very, very hazardous, and we have to prove to not just to our safety department, but to, say, the CDC that our inactivation methods are sufficient so that when we sterility test, things can come out for us to actually analyze. And we have to have different fixatives for different types of procedures, because we need to do X-ray analysis. We can't use specific buffers that might have, arsenic versus buffers that won't have arsenic. And of course, we want to do elemental analysis. We can't use osmium tetroxide. And forensics typically does call for a short turnaround time. And we've validated our normal old-fashioned procedures so that we can go from receipt of sample to, say, a negative stain virus image in less than 5 hours, from receipt of sample to a traditional scanning electron microscopy image with dehydration and processing and coding in about 12 hours, and a traditional TEM image we can get in just under 24 hours. So we've worked at lots and lots of different protocols, and we've fine-tuned them, and they work for us the way we've developed them. So this is actually the image that I made for the abstract for this talk. And top left, you have the Bacillus anthracis, which makes the anthrax spores, and SEM here, and TEM right here. All of these organisms on this thing are hazardous to our health, okay? This is actually a picture of an Ebola virus in panel K here. And you typically see Ebola virus on the internet. They're like shepherd's crooks, or they're commas, or they're semicircles, or something like that. But when the actual the tissue culture or the cells in the body are producing Ebola virus, perfectly, they come out as little perfect pretzel sticks, okay? This is actually a single tissue culture cell that was infected with an Ebola virus, the Ebola geckos virus. It's been renamed, but I prefer the word geckos because it just sounds kind of cool, okay? All these little squiggly guys you see right here on the side, these are virus particles that would have budded out of the cell if we had not inactivated and sterilely tested the cell so that we could image it. But if you look at the surface of the cell and you see all the wrinkles on the surface, these are actually virus particles moving laterally toward the cell, budding out, going to bud out from the cell in that direction. So this one cell right here will produce thousands and thousands of infectious virus particles. This is actually a Marburg virus. This is something called Rift Valley fever. So we look at lots and lots of cool things, right? This is our newest things right here on Q and R, right? So Q is a TEM and R is an SEM. So if you look at Q, this is the cytoplasm of a tissue culture cell in gray. In green right here, you have a lipid vesicle that contains what we now call the SARS coronavirus 2. Blue, you actually have the viral core, and yellow or gold, you have the viral spike proteins. So this is the SARS coronavirus 2 inside a cell. And R, you have tissue culture cells from the surface. You're looking at the surface of these cells, and the tan cells / gold cells right here. These cells are pretty much uninfected. The green cell here is heavily infected with SARS coronavirus 2. And all the blue / purpley things you see budding out of the surface and rebinding to the surface of this cell and to the surrounding cells are the SARS coronavirus 2 particles as they're being released. So CDC regulations. So the images here have nothing to do with this, but they're pretty pollen pictures, and I just wanted to put them here for you to see. Right? So fixation. We typically use something called a universal EM fixative, which is 4% paraformaldehyde with 1% glutaraldehyde. And we actually have to validate the fixative for each individual hazardous organism. So we have to take, say, Bacillus anthracis, we have to inactivate it with this fixative. And we have to then take the inactive material. So we say fix, say 6 tubes of Bacillus anthracis. We can simply call it anthrax if you want. So we take and we fix 6 tubes of anthrax. 3 of those tubes, we then have to sterility test to prove that they are sterile. The other 3 tubes we have to take and spike with Bacillus anthracis cells to prove that the inactive material that you just made does not inhibit the growth of those cells that could technically be damaged or injured that might still be there. So we have to sterility test 3 samples and then spike 3 samples and prove that they do not inhibit the growth of those cells that you are trying to inactivate. And this has to be done in triplicate by 2 different people. Once we've done this and we prove that the actual samples are sterile and that the sterile material we produce does not inhibit the growth of that organism, then in the future we can sterility test, we can inactivate things and sterility test, say 1/10 of that sample, and then the remaining 9/10 can be removed from containment for us to begin our processing and analysis. So this is for any type of hazardous organisms like anthrax, Yersinia pestis, Brucella, Burkholderia, tularensis, or any viruses like Ebola, things like that. So we have to, for each one of these things, do a complete validation of inactivation for that specific organism with that fixative. Very time consuming, very annoying, but it is part of CDC protocols. So again, these pictures are not anything to do with this, but they're just pretty pollen pictures, and I thought you might want to see what pecan pollen looks like because it's actually kind of cool, okay? SEM and light level, and this is actually saffron pollen and cherry pollen, okay? So the hardest thing for us to actually inactivate are bacterial endospores. We look at lots and lots of bacteria, we look at lots of viruses, and bacterial endospores are the hardest thing to inactivate. So this is what we set our baseline with initially. And it doesn't matter all the work that we've done in the past, we still have to go back and revalidate every protocol with that fixative. So samples that we get that may dissolve in aqueous fixative, we had to develop solutions for that. So we have different types of inactivations, right? So we have samples that may be fragile. So we have to have non-destructive analysis. We have to have samples with unknown organisms. So we have to prove that samples are sterile no matter what might be there. So we had to figure out the inactivation time for the hardiest type of organism, which in this case, are bacterial endospores. And bacterial endospores are resistant to heat, radiation, and to most types of chemical treatments. And different kinds of endospores are bacillus endospores. They're most all bacillus species produce endospores, Bacillus subtilis, Megatherium, anthrax, lots and lots of different bacillus species. And the Clostridium species, most people have heard of Clostridium botulinum, which causes botulism poisoning, Clostridium tetani, which causes tetanus. And this is one of the most common in hospitals. It's Clostridium difficile, and this is a common nosocomial infection. If you go in the hospital and you have to have a serious course of antibiotics, it will completely remove your endogenous or your endemic bacterial flora in your gut. And if you have any spores of Clostridium difficile that are in your gut, they will then hatch out, start growing, and you'll have a nice, painful Clostridium difficile infection. When we first started doing this, we took lots and lots of different bacterial spores, and we tried to figure out how long did it take to actually inactivate these spores. So we started thinking, well, anthrax has to be the hardest one to kill. Okay, so we actually took all these anthrax samples, which is 1 through 4. 1 through 4 actually anthrax that actually causes disease. The fifth one here, Bacillus anthracis stern, is actually the vaccine strain that is used to vaccinate cattle. Okay, but if you look, all the anthrax spores look completely dead in 30 minutes. And we're starting with over 10 to the 10 cells or spores per mil. We're talking with a very thick, not pudding-like, but pretty thick slurry of cells that we're inactivating. But if you look way over here, this guy right over here, this guy takes 4 times as long to kill than anthrax. And this is simply Bacillus subtilis. So we base all of our data on this Bacillus subtilis, which is much, much harder to kill than anthrax or Clostridium or things like that. Bacillus subtilis is even harder to kill than the thing that you use to validate autoclaves with, Geobacillus stereothermophilus. Okay, so this 240 minutes that we use is what we decided to use for all of our organisms. So we chose to use inactivation. We take our samples, we place them in our 4% paraformaldehyde with 1% glutaraldehyde for 240 minutes. And then we develop our validation of inactivation protocols with this time period. So in the future, we have to inactivate everything for 4 hours. Then we can sterility test it. Then we can actually bring it out of containment to begin our processing. In addition to spores, we also looked at live bacteria. So we looked at Francisella tularensis, which is commonly causes something called tularemia. It's also known as rabbit fever. We looked at Klebsiella pneumoniae, which a lot of people know causes a lot of different types of pneumonia. Listeria, which causes listeriosis, and Salmonella, which is a GI disease as well. But if you look at this, you got 1 times 10 to 11 cells. I mean, a very pudding-like consistency, very thick slurry of cells, and all these cells completely dead in 5 minutes. So you can definitely see that spores are much harder to kill than vegetative cells. So even with that, we decided to choose the 240 minutes as our typical inactivation time frame. So things that we've looked at and we've inactivated in 240 minutes or less. We've looked at the Bacillus anthracis, lots of different isolates of that. Bacillus cereus, which causes, if you're immunocompromised, can cause disease in humans, and all kinds of Bacillus species, and Brucella, Burkholderia, and lots of different viruses. Cowpox virus, Ebola virus, Francisella tularensis, Hunan virus, Lassa virus, Marburg virus, rickettsial species. These are tick-borne diseases, Rift Valley fever, which infects cattle, humans, and a lot of different other organisms. Venezuela equine encephalitis virus, also known as V, another Rickettsia prowazekii, and Yersinia pestis also, causes the Black Plague. So we've inactivated lots and lots and lots of different types of organisms in 240 minutes. And we first had to figure out, well, if you leave these things in fixative for 240 minutes, does the ultrastructure change? So we did a time frame initially from 30 minutes. We actually started with 5 minutes. We did 5 minutes, 10 minutes, 30 minutes, 1 hour, 2 hours, 48 hours, 72 hours, and up to 7 days. And what we determined is that as long as you leave the sample in fixative, it is fine. Okay, up to 7 days, none of the ultrastructure changes whatsoever. And we figured that this actually works for months and up to a year or so. After a couple of years, the actual fixative starts to polymerize and it gets kind of gluey, but the ultrastructure of the sample is still pretty much intact. So we looked at lots and lots of different things. So here, instead of looking at just spores, we looked at vegetative cells as well. This is Staph aureus, Bacillus anthracis. We got Bacillus suis, Burkholderia species. This is Burkholderia malii, and we have Bacillus megaterium. So the ultrastructure of these in 240 minutes is perfectly preserved. So, and we are something known as an ISO 17005 accredited facility. I hope you don't know what that means. I mean that explicitly because being ISO accredited means that you do way more paperwork than actually lab work, okay? So when we're ISO 17025 accredited, what this means is that each sample that we get, when we process it, each image that we take looks like something like this. So this is sample number NB14001. So this was sample from NBAC in the year of 2014. It was sample number 1. And this digit right here, the 01, this is either the block number or the stub number or the slide number, okay? And the last set of digits here, the 03, this is the actual image number. So each picture that we take has an individual stamp that looks like this and a timestamp. From this number right here or the timestamp, I can pull every training record from anybody that was associated with the processing of that sample, all the equipment records to prove that all the equipment was in scope or it had, it's received its yearly PM or it's working properly, that it was not out of compliance. All the chemical records to prove that all our chemicals are not expired and that they have been approved for release. So that when we get reagents in, we actually have to lot test them to prove that they are acceptable for use, okay? And all the paperwork for every bit of this has to get uploaded to this digital program called LIMS, Laboratory Automated Information System, and so that the clients can access this real time. So when we do this, since we're ISO accredited, all the images are very reliable. All images from the same samples are rated so that they're rated on a scale from 1 to 10. And any image is actually usable because if you get a really, really crappy sample, a forensic sample, I won't say something that looks, gross, but we can get very, very nice images from samples that are very degraded. So the sample may be degraded, but our processes allow us to get a very, very good image of that degraded sample. Okay, it won't be pretty sample, but it will be a very, very pretty image of that degraded sample. And we process each sample under the exact same conditions because we have the database that we've developed called the Bioforensic Image Reference Database of all these hazardous organisms. And if we need to compare anything new to it, we have to process it the exact same way so that the client can have confidence that the images we produce now are the exact same as the images in the database, okay? So here, these are not all hazardous organisms here, but on the bottom left, you have a Rhizopus stylonifer spore, and you may say, well, what does this spore have to do with anything in forensics? Well, we look at lots of different kinds of pollen samples because forensic samples, the clothing you have on right now has pollen on it from where you live at. And if you go online, you can follow the allergy database to figure out which pollen samples are high, which parts of the month, and which times of the year. And if you vacuum your clothing, we can find the pollen to figure out, in general, the region you actually have been in or are from. We have colleagues, Department of Homeland Security in Chicago, which can take like the air filter out of a truck and tracing the pollen from the outside of the filter to the inside of the filter, trace that truck route back to where it came from using the actual pollen inside the air filter, which is pretty cool to me. I thought it was cool. The second image here, Ricinus communis, this is simply a broken open castor bean, and this is most people's favorite image that we produce for some reason. It's very Christmassy looking. So this is actually a single cell inside this castor bean. The green stuff is cell wall material. The yellow gold stuff here is castor oil, also known as ricillinic acid. It's an excellent industrial lubricant, which is why we grow these castor beans. But the red stuff is things you do not want from castor beans. So the red things you see here are actually crystals of ricin protein. And a lot of you have heard of the word ricin because people try to make and purify this stuff. And I can simply look at a sample of ricin, and if I can see that the lipids and all the stuff is still there, that these people have just not really begun going down that rabbit hole of a nefarious act, they simply took a castor bean and crushed it. So the third thing is rickettsia, proezechia, tick-borne disease, and the fourth is more pictures of anthrax. Okay. So ISO accreditation. What does it mean? What is it good for? It means that everything that we do will eventually go to court because somebody is going to go to prison. So we have to be able to prove that everything we do can stand up in a court of law. So if you look at the ISO 17025 here, not only do we have training and reagents and all this stuff all over here, but we have to actually do competency testing of each person that's doing the actual procedure. And I have to write an authorization memo authorizing them to do this procedure. We also have to do method validation for every single method we do to validate that it works in this space. So it doesn't matter if I take a paper and show that people have done this elsewhere, I have to repeat this in the actual laboratory I'm in to prove that it works in this space. And every few years, we have to do something called proficiency testing, which means that not only can I do this procedure, but in random, anybody that works for me can do the procedure, proving that not me, but the laboratory itself is proficient at doing this actual procedure. And we have quality controls. And I hate the word quality controls, but because we have a big quality department, and this quality department has lots and lots of things they make us do. Before we can actually use a light microscope, we have to take a stage micrometer and document that the microscope is working properly. Before we use a scanning electron microscope, we have to take, say, a Geller MRS5 or MRS6, because these are ISO accredited, and we have to prove that the microscope is interpreting the magnification correctly and that the microscope is putting the correct size scale bar on the images before we can take any pictures. If we're doing X-ray analysis, we have to take copper, and we have to prove that the detector is working properly by ensuring that the K line is at 8.04. And we have to quantitate something to prove the software is actually interpreting what the detector is proving that it's working properly. So what we do is we use a NIST stainless steel, and we look at the amount of manganese inside the stainless steel to prove that the detector is interpreting the sample properly. So all these standards that we use for quality control have to be either NIST produced or NIST accredited. And if you don't know what NIST means, it's a National Institute of Standards Technology. It's in Gaithersburg and other places in the country. So I have lots of stuff that I do that I can't talk about. So I basically just show you pictures and talk about things that I can talk about. Things that we actually looked at before we were able to look at hazardous organisms include things like this bee stinger that we found dead in the stairwell. This fly was actually in a spider's web, and it's kind of cool that the eye is actually missing on the spider. That was just kind of very cool, I thought. An insect antenna, a milkweed siphon here, and a stink bug that my boss back then brought to me because he wanted to see what the stink bug head looked like. And these are just different types of microscopes I've used in general and different types of samples that I have looked at. So I'll be happy to answer any questions you guys have. Again, I look at lots and lots of different kinds of things. I've been doing this since basically 1988. And if you have questions, I'll be happy to answer them as best I can.
Thank you, Robert.